Welcome to the University of Rochester Pathology video series. This video introduces the basic histology of the spleen. The slides shown in this video are available from the University of Rochester's digital slides collection, slides UR H094 and 095. The spleen is a vascular organ consisting of a network of capillaries, sinuses, and lymphoid follicles or nodules. The very outside of the spleen consists of a thin layer of mesothelial cells. This thin layer surrounds a thicker fibrous layer of connective tissue and muscle cells known as the splenic capsule. Together, the mesothelium and capsule form the outer surface of the spleen. Some muscle tissue extends from the capsule wall deeper into the spleen, forming trabeculae that interface with the inner pulp. From these trabeculae extend trabecular arteries, which diverge into central and peninsular arteries, which are associated with the two main types of splenic tissue, red and white pulp. Central arteries feed the spleen's white pulp, and branch off to form peninsular arteries, which feed the spleen's red pulp. A majority of the spleen is made up of red pulp. Red pulp consists of a network of blood vessels that act to filter and store blood and its constituents. Specialized capillaries called sinusoids have specifically sized gaps in their vesicle walls which act as a filter. As blood flows through the sinusoids, red blood cells are squeezed through the vascular gaps. Old or unfit red blood cells that are damaged by the filtering process are consumed by the abundant macrophages and other monocytes that reside in the red pulp. The white pulp consists mostly of lymphocytes and other lymph tissue. Generally, lymphocytic T cells aggregate and form para-arterial lymphoid sheaths that surround the spleen's central arteries, whereas lymphocytic B cells form lymphoid follicles that form adjacent to and stem off the central arteries. These lymphoid follicles consist of a lighter stained germinal center and a darker stained corona. The germinal center contains mostly proliferating B and plasma cells. The darker stained areas consist of a majority of non-proliferating B cells and some other lymphatic cell types, such as T cells. Note: without cell-specific staining, B cells and T cells cannot be easily distinguished. The area of cells surrounding the corona is considered the marginal zone and separates the red and white pulp. As the spleen does not contain any lymphatic vessels, all lymphocytes produced in the spleen enter the body traveling through the vessels of the red pulp. In summary, the spleen consists of a protective fibrous splenic capsule, venous red pulp, and white pulp. Red pulp is populated by red blood cells and monocytes, and is the gateway for lymphocytes into the bloodstream. White pulp consists of lymphoid cells that form para-arterial sheaths and lymphoid follicles that contain mostly B cells and also some T cells.